This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 882, recorded on March 29th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi. It's great to be here. It is cloudy and 35 Fahrenheit. Winter has made a little bit of a comeback recently. We had snow even yesterday.
It's freezing.
Yeah, it is.
It's freezing here. It's totally crazy. It's 1 degree and cloudy here. I had to break out my old winter coat, but I think it's going to warm up, which is nice.
I think so too, and hopefully this maybe will be the end of winter.
Yeah, that would be good. I'm ready for a little warmer weather.
Me too.
And today, today we have just me and Brianne, which is we've done this before, I think, right?
Yes, we have.
And so I picked a couple of immunology heavy papers. So I hope you enjoyed them. The first one, our snippet actually was suggested by Bonnie Offit.
I like seeing that.
This is our second email from Dr. Offit, who is a pediatrician and the wife of Paul Offit, of course. She wrote, "Hi, Vincent and crew. I would really love to hear you guys toss around this newly published commentary. Seems spot on to me as we continue to see waning neutralizing antibodies in the setting of epidemiological evidence of persistent protection against severe disease after vaccination and or natural infection in the vast majority. Perhaps the mRNA vaccine technology is even better at inducing strong T-cell immunity than we realize. These authors certainly make a good case that measurement could serve as a better correlate of immunity. The authors suggest that at the very least, we need more research in this area of T-cell memory immunity." And she links to the paper we will talk about. Thank you, as always, for your amazing podcast. Please keep up your great work for all the fans like me. It is signed Bonnie Offit, pediatrician and Paul's wife. And I had seen this also and thought it would be fun to chat about it. It is a commentary, right?
Yes, and I agree. And I think this is very much up my alley. I got to think a lot about things I spent lots of time doing in graduate school. And it was really nice to see so many findings we've seen about T cells put into one place and put into context together. And
you have to admit, Brianne, we've been saying T cells for 2 years now, right? In terms of COVID.
We have,
and
yes.
So now it finally gets into the mainstream journals.
Right. Well, and I also liked the section of this where it talks about the issues with measuring T cells and different responses and things like that. That it was nice to feel a little vindicated that we had been on the right track.
And so it is in Science Immunology. It is the title is Understanding T-Cell Responses to COVID-19 is Essential for Informing Public Health Strategies by Santosha Vardana, Lance Baldo, William Maurice, and John Werry from Memorial Sloan Kettering here in New York City, from the Mayo Clinic, from the University of Pennsylvania. So this is, I suppose, what is it? A review article or an opinion piece or a combination of both? I guess
I kind of think of it as a combination of both. It reads to me mostly like a review article, though perhaps it's more of an opinion and I just didn't need to be convinced.
Yeah, I think us on TWiV don't need convincing. It's called a viewpoint. Okay, That's what it is. But as you know, I think I told you a while ago, a well-known B-cell immunologist wrote me a letter saying there's no evidence that T-cells play any role, which I think is just not right. But
yeah, and that's what I really liked about this is from now on, I'm going to have a source that puts a lot of that T-cell data together. Right. And I think that even some of the people who downplay the importance of T-cells will note that some things that they talk about are cited in here. And I think that some of it can be a little bit disentangled nicely.
Yes. So we'll go through some of the high points and chat about them as we always do. And the first point they make, so there's subheadings. This subheading is called Cellular Immunity Plays a Crucial Role in Resolution of SARS-CoV-2 Infection. And they note that infection, of course, induces both humoral and cellular immunity, and much of the focus in vaccine development has been on neutralizing antibodies and less on the role of T cells and, Brianne, non-neutralizing antibodies.
Yes, they do a nice job about talking about things that antibodies can do other than neutralization.
And this so echoes what we have been saying, not because we're prescient, but that's science. Science is to look at all things at once, right?
Right. And, there are certainly many novel things that we have seen during this pandemic, but there are also an awful lot of ways that this virus is following the rules that we've seen in textbooks for a very long time.
And so they conclude in this little paragraph, mounting evidence suggests T cell contributions to the host immune response are required for early, broad, and durable protection, especially in the setting of new variants of concern. Everything is referenced here, by the way. It's not just their opinion. They have multiple references after each statement. The next section or subsection, T cells can recognize a broad range of SARS-CoV-2 antigens, and they make the point that T cells recognize a broader range of viral epitopes than do antibodies, and so they may be better at recognizing infection by variants of concern and otherwise. And they say that they can recognize areas that are not subject to mutation-driven escape from antibodies, which is...
They also mention a little bit, and I think it's important to point out that at this point in the viewpoint, they're really talking about infection, and they're not talking about immune responses to vaccination. They talk about in the case of infection, you also can really see good T cell responses to many parts of the virus other than spike. It's not just about spike.
Then they go on to talk a bit about T cell epitopes. They say T cell epitopes are shared among variants. On the average, spike-derived epitopes are conserved at 100% amino acid identity. These constituted 84.5% and 95.3% of the total CD4 and CD8 epitopes. And they say there was no difference in the CD4 and CD8 T cell response to delta spike peptides compared to ancestral. And in contrast, of course, we know that the variants are evading humoral immunity. And that's why I think the most important reason for knowing that is because if you want to treat someone with a monoclonal antibody, you want to make sure that the variant currently circulating is not going to be resistant to it.
Right, absolutely. And I think that this, gets at some questions about how worried we should be about some aspects of the variants and shows that, while yes, our antibody response is changing, the T cell response does seem to be going to protect us potentially.
They note that sera from vaccinated and convalescent individuals have a 4 to 6-fold decrease in neutralization against Delta, which we, of course, have covered as well. Monoclonals have reduced neutralization. Delta, Omicron, Alpha, Beta show all of this. And Omicron, of course, has considerable antibody escape. And yet they conclude, despite the loss of all of this neutralization capacity, vaccine efficacy in protecting from severe disease, hospitalization, and death has been only minimally to moderately affected. And they say, hey, maybe this is durable cellular immunity. And they say in support of this, and that is a theme we repeat all the time, isn't it, Brianne?
Yes, exactly.
70 to 80% of the CD4 and CD8 T cell epitopes in spike are not affected by Omicron, and T cell responses are largely preserved. Mostly, I think, the work of Crotty and Sete and a few others, right?
Yes, exactly. And so it looks like, with infection and subsequent infections, you are sort of having great T cell breadth that's going to protect you against many different variants.
The next section is called T cells protect following reinfection and provide durable immunologic memory. And then I like this part here. They say our limited understanding of the waning immunity has contributed to implementation of immunization boosters. It's so true, right?
Yeah.
For some reason, as you would say, Brianne, people forget their basic immunology, that antibody levels contract. And just today, the FDA approved a 4th shot for 50 and over.
Yeah, I saw that. And I was receiving a lot of texts today asking about my opinion on that. And I felt like my opinion was perhaps too long to respond by text.
No, and I bet if you asked Paul and Bonnie Offit, they would agree that there seems to be little justification for that.
Right, exactly.
On the other hand, reports of association of optimal T-cell responses with mild disease following infection are supported by more recent accounts of protection conferred by memory T-cells on secondary exposure. T-cell responses have been found in individuals who fail to seroconvert. So seroconversion means making antibodies following asymptomatic or mild COVID.
And that's super important in terms of thinking about epidemiology and thinking about how many people have been infected.
Right. Yeah, I think somewhere in this article, they point out that we're probably underestimating infections by seroprevalence because not everybody seroconverts. Yet they're still developing only mild disease. And they point out people who get B-cell depleting therapy, of course, for various reasons, they have a decreased antibody response, but their T-cell response is not affected by that. And they conclude that a consensus is evolving around the significance of high levels of neutralizing antibodies in mediating protection against infection, with a complementary role for T-cells and memory B-cells in preventing severe disease and hospitalization. And so I would agree, except this protection against infection is only going to last as long as you have high levels of neutralizing antibodies, which is 6 months, right?
Right. And so, that gets back to this whole idea of what are we actually trying to protect against with our vaccine? And that should dictate what goes on with boosters.
So the booster is clearly an effort to keep high levels of antibodies.
Exactly.
And so if that's the case, then Offit pointed that out in his Philadelphia Inquirer opinion a while ago, then we're going to have to boost every 8 months, essentially, which seems not, as we've said many times, it's just not a good strategy.
Yeah. And so we need to decide, are we really trying to prevent all infections by keeping antibody levels high? That doesn't really seem like something that makes a ton of sense. And that's not what we do with most other viruses.
Not at all.
Or
not at all. No.
Are we trying to protect against disease, in which case we don't necessarily need those boosts?
Yeah. They talk about the durability of the T-cell response, right? How long does it last? And we don't really know because it's only 2 years now. But they say robust CD4 and CD8 T-cell responses have been found up to a year post-infection, 6 months post-vaccination. And for SARS-CoV-1, memory T-cells were found 17 years after exposure. 17 years, SARS-CoV-1. So if that's the case with SARS-CoV-2, and we'll have to wait 16 years to find out.
I know. And there are some other papers out there looking at the longevity of some other memory T-cell responses that come up with some pretty dramatic numbers. When I read this paragraph, I realized that my heart went out a little bit to some of the graduate students who were interested in this problem, because to be a graduate student interested in this problem, you need to do some long experiments. And as a person who had mouse experiments that went out to 300 days to look at durability of immune responses in her dissertation, I feel for those graduate students who are trying to answer this question.
Well, the good news is mice don't live 17 years.
That's true.
You can 2 to 3 years it works, but there's still a long time in a dissertation period.
It is.
Yeah. As the next section is called T-cell activity in response to SARS-CoV-2 may signal anticipated disease severity for patients with COVID.
I really like this section.
So they say that, so the idea here is that our insights can give us an idea of the role of T cells in understanding the outcome of infection. And they say, for example, the early induction of interferon gamma secreting T cells. So, correct, Brianne, interferon gamma is often a marker for activation of T cells, correct?
Yes. So it's often made by CD4 cells and CD8 cells that are particularly good at fighting virus.
And so a naive T cell wouldn't make it, and then as soon as it sees antigen,
it will start making interferon gamma.
And then you can assay it by like an Elispot assay, right?
That is correct.
And they say limited mapping of the epitopes have revealed certain viral proteins have dominant epitopes shared by multiple individuals. And they say we should really define these, and particular what HLA molecules are recognized or presenting these epitopes. And so maybe we could say, oh, you have this HLA type, you're at particular risk for severe disease, right? HLA polymorphisms and severe disease.
Yes. And we can say that with a number of other viral infections because of this work having been done.
And in fact, the more severe disease in some studies is associated with lower frequencies of CD8 and CD4 cells. In one report, they found that specific peptides were enriched, raising what they call an intriguing possibility that the immune response to certain epitopes may contribute to disease severity. What did you think about that, Brianne?
I think that could make sense in some situations. We know that sometimes we can see immunopathology. And I think we don't necessarily know enough about all of these details to say that we can't see a situation where sometimes T cells are contributing to disease. What I will say that I think is important here is that this viewpoint is nicely divided into a section on immune responses following infection and a section on immune responses following vaccination. And right now we are in the section looking at immune responses following infection. It is possible that in a situation where there are T cells induced by infection that are maybe coming in after we've already seen some other damage from the virus, some other inflammatory damage or something like that. If those T cells happen to be against the wrong epitopes, we might see some damage. I think that that's possible. But I don't think that tells us about T cells elicited in the vaccine case that are present before infection. And so I like that they bifurcated this here.
So the rest of this piece is really about measuring T cells and what the consequences that would hold for public health. And they say that we really need to understand T cell responses to understand the immune response. We have techniques to do this, and they go through them. For example, Elispot for measuring interferon gamma, intracellular cytokine staining, ICS, activation-induced marker, AIM assays. So I know Elispot and ICS. What is AIM, Brianne?
Honestly, their discussion of AIM made me laugh a little bit because that was something that I always knew as immunophenotyping or just doing flow cytometry, and now it has a fancy name. So basically, this is using your flow cytometer to look at whether or not cells have markers associated with activation.
Right. Yeah, we've done a couple of papers where they use that. Yeah. And they also talk about using tetramers to stain for specific virus-specific T cells. They mentioned that the FDA issued an EUA for a high-throughput assay that relies on sequencing, next-generation sequencing, to characterize T cell receptors. But they say, these are great techniques, but everyone has an advantage and a limitation. And one of them is, the advantages of all of these, and especially next generation sequencing, T cell receptor sequencing, you get a lot of information, but they are costly and they require very highly trained personnel. They do point out that it's much easier to do a neutralization assay.
Yes, exactly. If you want to do most of these T cell assays, if you want to do ICS or the AIM assay, you need a flow cytometer. You need someone who can use that flow cytometer and analyze the data. In all three of those assays, you need to have cells from the patient. So you need to either have frozen cells or live cells from the patient instead of a serum sample.
Right.
And that actually has often been a challenge to get enough PBMCs that are viable to do all of these assays.
Right.
And, that's possible. Even with the Elispot, you need those cells. And the TCR signaling information here, or the TCR sequencing information here, is amazing, but also involves so much data.
Yeah, they make the point that we can make overlapping peptide pools now, which is very commonplace, cover all the potential T cell epitopes, but you need, as you say, fresh cells to add these to from patients to look at stimulation, and that's often hard to do. Fresh blood samples or cryopreserved PBMCs that have the ability to respond to these. And they say this is a key point. These issues present significant barriers for the use of these assays in the clinical lab environment, which is where a lot of these assays will be done, a lot of neutralization assays done in the clinical environment. Not often easy to do that. Right?
No, because you can get a sample, an antibody sample with some serum pretty easily to do some neutralizing antibody assays. But separating out your PBMCs, getting a number of PBMCs that allows you to do more than one assay or look at more than one peptide, all becomes really complicated.
So they point out that many of these approaches are predominantly investigational tools that you would use in a research laboratory. And they say we need to have some kind of framework for saying, first, which of these should we put in the clinical lab and how? And that's really important. I mean, that's like, that's a thing we don't often think about. We just, in the research lab, we do our experiments, right? But a clinical lab is a different story. So I think that's very important. And it's not going to happen overnight.
No, not at all. And how do we then make sure that everyone in the clinical lab is trained in the use of these assays?
They talk about a couple of interferon gamma release assays that have been developed, like Interferon for SARS-CoV-2. They haven't got FDA authorization yet. And according to this, in November 2021, the FDA said, I'm not going to permit any test to be marketed without first being reviewed and authorized under an EUA. For a while, they were letting them be done, and then they changed that.
Yeah, I found this section a little confusing, where they talk about these two tests that have been FDA approved for testing for TB, two different tests. And they say that they have, where is it?
Replaced the PPD.
The PPD.
Yeah.
That surprised me. I think I've had a PPD in the past couple of years. I'm not sure if my PPD has been replaced.
I was surprised to hear that, and I didn't hear that it had replaced it, but it would be interesting if it has, right?
Yeah. Oh, no, that would be fantastic, and I would really like it. I don't enjoy getting a PPD test, but...
So an interferon gamma release, what would that involve? Taking a little blood and then adding peptides to it? And looking
it'd be taking a little blood, adding some peptides, and then looking for the interferon release.
Interesting. Then they talk about how most population-based surveillances are using seroprevalence in blood donors, which is antibodies, neutralization, and so forth. But they say there's no big programs to analyze T-cell responses at a national level. I mean, think about this. Millions of samples are being analyzed for neutralizing antibodies, nothing for T-cells at the same level. And they say we need to fix this. Because, first of all, as we said before, the seroprevalence is probably wrong, and we need to know about the T-cell responses. So I think, Brianne, the point here is that you have to decide what you're going to measure, right? Everybody can't be measuring a different thing.
Right, exactly. And we need to think about things like how we're taking samples in terms of making sure we have PBMCs or something like that. Because earlier throughout this, there are some interesting questions that are brought up, and we just don't have the data to get further answers on them.
Yeah. They also have a section here where they point out some of the shortcomings in the vaccine trials. They say, we're really only looking at antibodies. The FDA only requires outcome data, right? Infection or disease. They don't say you need this amount of antibody. Of course, they don't say you need this kind of T cell or this level of it. And they point as an example, the Pfizer vaccine trial only used antibodies in the analysis. And they say this failed to account for the possibility of a protective role by vaccine-induced T cells. In other words, they said, oh, the antibody levels are not high enough. We're not going to go forward. But that seems to me, yeah, they're right. The T cells might have been induced and you just didn't know it.
Right. And you know, even in some of the early data on the Pfizer vaccine for adults, there was a figure or two of T cell data. Specifically, I remember looking at Th1 versus Th2s and seeing Th1s and seeing a little bit of CD8 data. And so you could do some comparisons even with that and this pediatric population. Yeah. But that was not done.
So they say in the future, vaccine trials should require assessment of both cellular and humoral immunity, which I think is a great idea. Right. I also think that if we had a better understanding of immunity, it would, they say, help counter vaccine information, which they say has arisen because we're not thinking about T cells. It's a TWiV theme, right?
Yes, yes.
They say a case in point that received media coverage, which suggested that the adenovectored vaccine, Ad26-CoV2S, was 50% less effective against viral variants. This was all based on antibody neutralization, but the T cell response was unchanged, and the recipients of the vaccine were protected against disease. But the press focuses on the neutralization because that's what they're fed, unfortunately, by the scientists.
Right.
And that's wrong. You have to, and I think only now are we slowly starting to see T cells in the news, right?
Only slowly, and with different and not always entirely correct discussions. And it's really kind of interesting because these types of vaccines, the mRNA vaccines and the adenovirus-based vaccines, have often been thought of as being particularly useful because of their ability to elicit good T cell responses in addition to antibody responses, since the viral protein antigen is being made inside of the cell. So it's interesting that we're now using these vaccines that are supposed to be the vaccines that also give you bonus T cells, but then we're not actually measuring the T cells.
Right. So they end up by talking again about how we're focusing on antibodies, but you don't actually have, and this may be what that immunologist who wrote to me was talking about, critical data supporting the role of T cells in actual protection is vital and currently lacking. And the ideal goal would be to define protective T cells following infection or vaccination, or I would say both, right?
Both.
And they
I agree both.
We need to have both. And they talk about a recent study where they had 3,000 participants who were measured by an Elispot assay and then followed up for symptomatic PCR-confirmed infection. And the conclusion was that individuals with SARS-CoV-2 reactive T cells were protected from COVID-19. And they say we need more of these studies, bigger studies like that, to give us an idea of what's important. I would say if we had more of those studies, we wouldn't be getting so many boosters.
Right. I agree. I think that if we knew more about what each administration of the vaccine is giving us, we would know that we were okay.
So the key is, Brianne, we need to measure not only in vaccine trials, but also when we do these big studies, not just measure neutralizing antibodies, but T cells. But you have to decide what aspect of T cells you want to look at, because there are a couple of assays you can do, right?
Yes, and you're probably not going to get enough cells to do all of those assays. So you need to prioritize.
Which one would you pick?
I would probably pick something like intracellular cytokine staining, because I could use it to look at multiple cytokines at once.
Right.
Instead of looking at one cytokine, as you usually do with Elispot. So with Elispot, you usually just look for interferon gamma producing cells. With interferon with intracellular cytokine staining, you could get the ability of those cells to make multiple cytokines. That's my first bias, but I could certainly see arguments in a few different directions.
So for the intracellular staining, you would take PBMCs from patients.
Yep.
And then you would add peptide pools to them.
Yes.
Is that
So
correct?
you would stimulate them with peptide pools, and you would look at the fraction of cells after stimulation that made interferon gamma, or that made TNF alpha, or that made IL-2, or that made 2 cytokines, or that made 3 cytokines, which are known as the multifunctional cells, and are thought to be the most useful.
Right. I remember that from a recent discussion. Or polyfunctional, right?
Yes. Polyfunctional cells are the best.
Okay. Anyway, so that is the viewpoint. I think they're spot on. They're Ellie spot on. Right?
They are Ellie spot on.
Well, that could be a title, Brianne.
It could be a title. I like that.
Ellie spot on about T cells. How about that?
I think that would be good.
Yeah. It's great. I like it. Thank you, Bonnie Offit.
Yes. When we used to do Ellie spots in graduate school, one of my fellow lab members really, really, really wanted to get a Dalmatian so that she could name it Ellie Spot, because she did so many Ellie spots.
That's brilliant. That is brilliant. Maybe I'll make that as the art for the thumbnail. That's very cool. Did she ever get a Dalmatian?
She did not, to my knowledge, ever get a Dalmatian, but she was ready to get a Dalmatian and name it Ellie Spot after the technique she did all day.
That's great. I love it. Many scientists name their pets after some of the things that they work on or do. I like that very much. All right, now we have a paper in Nature Immunology, and only with Brianne here would I venture to do such a paper, but it is really interesting. The name is IL-1 and IL-1ra are key regulators of the inflammatory response to RNA vaccines. And this is from a large number of authors from both BioNTech and Genentech, 2 companies. BioNTech, of course, the developer of the Pfizer mRNA vaccine. The first author is Siri Tatinen, and the last author is Ira Melman. And buried in there is Ugur Sahin, who's the guy at BioNTech, one of the founders. And Ira Melman, he's currently at Genentech. I knew him vaguely years ago. He used to be at Yale, and was a cell biologist. He was at Yale, not an immunologist. Very interesting. But
I know people he trained who were definitely immunologists.
Who did he train that you know?
So someone in my graduate program named Shannon Turley, who was one of the faculty at Harvard when I was there, had trained with him.
So when I visited Yale many years ago, when Ari Helenius was there, and Ari Helenius studied virus entry and endocytosis and so forth, and he collaborated a lot with Ira Melman, who was also interested in a lot of cell biology things. And then at some point, he moved to Genentech, and his interests broadened. So, and this paper is a reflection of that, which is a nice thing to see, because there's no reason to stay stuck on one thing your whole career, which many people do.
Yes, this paper made me want to run into the lab and do experiments.
You want to go and I'll wait for you?
No, it might take me a little while. Well, I will wait till more of my students are around, so I have some extra hands.
So this paper is going to begin to tell you why when you get those mRNA shots, you have a lot of reactogenicity. People have kind of anecdotally said, my arm is really sore. I had a fever. I felt kind of crappy. This paper is going to start to explain why that is. It's all about RNA vaccine platforms. And you know from listening to TWiV that these RNA vaccines are capable of activating the innate immune response. They're recognized by pattern recognition receptors, and that consequently induce the production of what we call pro-inflammatory cytokines, and you get fever, chills, and so forth. And no matter how you give these vaccines, you always get that, whether it's intravenous or intramuscularly, how they're formulated. And this is going to be important here. You can wrap RNA in a liposome or a lipid nanoparticle. Of course, the Moderna and Pfizer's are in lipid nanoparticle vaccines. And it doesn't matter if you have modified or unmodified RNA. So remember, unmodified RNA is a really good activator of toll-like receptors. And the cool finding of Catalin and Weissman was that if you modify the RNA with like N-methyl pseudouridine, it dramatically drops down the ability of the RNA to be recognized by toll-like receptors. Yet it will still, if you incorporate that RNA into a lipid particle, it will still activate pro-inflammatory cytokines.
Right. And it's also important to note, sometimes people are like, oh, well, if you put in pseudouridine, you get no activation. And I like how you said it dramatically reduces, because you do need a little bit of innate immune activation to help you get that good response from the vaccine.
Yeah, you just don't want to have enough, so much that the vaccine can't get going.
Right, exactly. You don't want to have, and you also don't want to have so much that your patient has side effects that are negative that make them not get that vaccine.
Now, here's the cool part. Mice get very few side effects from mRNA vaccines. And you may say, well, how do you know? Because, well, we can measure things. We can measure inflammatory cytokine release. You can give them intravenous liposomal vaccines with unmodified RNA. So this is called RNA-LPX. This is one of the things we're going to come back to. So unmodified RNA in a liposome formulation. You can give mice 50 micrograms of those, and they have moderate flu-like symptoms, fever and
those are well tolerated in mice.
Mice. And did you know this? Because I didn't know this.
I did not know this at all. And like I said, that's why I want to run into the lab.
And they say that even at these doses, 50 micrograms in mice, which mice don't care about, patients get mild to moderate flu-like symptoms. And they say this reduces our ability to explore doses. So we can't give higher doses to people because there's too many side effects, but maybe a higher dose would be better at making a better T-cell response. So they say, we need to know why this happens and see if we can get around it.
Right. And it also means that, that also tells us that maybe the response we're seeing in mice in general is a little bit different. And so maybe some of our things we've learned about these vaccines, maybe, we didn't know because of looking in mice that these vaccines were going to be as great as they are.
Yeah, well, in fact, that's the problem. Like in mice, we didn't see many side effects. And so then we put them in people. We see a lot of side effects. It was a surprise.
Yes.
Right. So now they say this is a great number. There's the doses in humans are more than 1,000-fold lower than in inbred mice. So that's the difference you can put in. You could put 1,000-fold more in mice and it doesn't affect them. But in humans, you're limited.
And I love that the beginning of that sentence is, given the obvious size differences.
Yeah, right. And you may say, well, I don't care if a mouse responds less to an mRNA vaccine. Well, you have to, because if you can figure out why, maybe you could do something in people. And that's why we study mice, right? To learn things we can apply to people. And so that's what this paper is about. We don't know why mice and humans are different in their response to these mRNA vaccines. So they decided to figure it out. And boy, did they. They did so many experiments. They did. And so they start with one cool thing. All right, so what are the factors that make these responses? They have a cancer vaccine, which I guess they've been working on for a while. It's an unmodified RNA in liposomes. Right. And so it's a cancer vaccine, and they take human peripheral mononuclear cells, take them out, put them in culture, and then they add this RNA-LPX to them. And they see a lot of cytokines made, a broad range of cytokines, including pro-inflammatory cytokines. It seems to be that monocytes are they do an experiment showing that monocytes are very important for the production of these. And one of the cytokines is IL-1 beta. Yes. So, Brianne, what is IL-1 cytokine? Tell us some cool things about it.
So when I think of IL-1 beta, I think of a cytokine that's really important in particularly things like fever. So it's a pyrogen.
Okay.
And it's a big part of kind of the early innate immune response. It is frequently made in response to a lot of different bacterial infections. But compared to many other cytokines, it's also unique in how it is made by a cell. So most cytokines are transcribed like any other gene, and then their proteins are secreted, sort of the way you normally think of a secreted protein. IL-1 beta is a little bit different. So IL-1 beta is transcribed as an inactive, transcribed and then translated to become an inactive protein. And so it requires a second step in order to be generated. It requires the cleavage by a protease, a caspase called caspase-1, which once upon a time was called ICE, or IL-1 converting enzyme. So this second step will cleave the inactive IL-1 beta, pro-IL-1 beta, into active IL-1 beta. And then there is secretion of that active IL-1 beta through a unique type of secretion process, because it's coming from the cytoplasm, not from different vesicles in the cell. And so this cytokine is not the only one, but it's sort of the one they talk about in this paper that has this mechanism, requires this special two-step pathway to allow its production and regulation. And when it is produced, it's a potent inflammatory cytokine. You can imagine that it's kind of nice that it takes two steps to get activated, given that it is so potent.
Yeah. So it's caspase. One step is caspase cleavage, and the other step is the inflammasome.
No. So the first step, it's known as signal 1, and that's just transcription.
Okay.
And signal 2 is the inflammasome, which is basically caspase-1 getting turned on. So the inflammasome you could also think of as caspase-1.
Okay. So they do experiments to show that both using inhibitors, that both caspases and the inflammasome are involved in the induction of IL-1 beta by this RNA LPX cancer vaccine.
Yes.
And so they want to know what parts are doing it, what parts of the vaccine are doing that. And so they found that the liposomes alone without RNA can give one signal.
Right.
Signal 2, which is caspase cleavage, I guess, right?
Exactly.
Activation of the inflammasome, right?
Yep. So the RNA is responsible for leading to transcription of pro-IL-1 beta, the precursor, and then the lipid activates the caspase to cleave that and make the active IL-1 beta. Right.
So if they modified that RNA, then they get a lot less IL-1 beta produced.
Right. Because
that's one of the two signals, right?
Yeah. They got rid of signal 1, so it doesn't really matter that signal 2 is still there because signal 1 was reduced.
And so if they do that, or if they put liposomes without RNA, they also diminish the IL-1 beta because they're taking away the other signal.
Exactly.
Right? Very cool. It's so nice that they can do all of these things. They have a lovely RNA liposome component, and they can take away and say exactly what is doing what.
Yeah, and they also have some really nice inhibitors where they can also use drugs to inhibit different parts of the inflammasome pathway to kind of also show the specificity of that pathway here.
So, for example, and building on that, they have a TLR7/8 agonist, right? So TLR7/8 would be activated by the unmodified RNA, right?
So that's signal 1.
And if they only add that, they don't get IL-1 beta.
Right.
Because there's no signal 2. But if they add liposomes with it, then they would get production of IL-1 beta.
Right, exactly.
So R848, which is this agonist, plus empty liposomes or modified RNA, they get IL-1 beta production. The other thing they say is that this RNA LPX, when they add it to cells, generates mitochondrial reactive oxygen species. And this is for monocytes when they add them to monocytes. If they inhibit that, then the IL-1 beta goes down.
Right.
So tell us about mitochondrial reactive oxygen species in monocytes. What would be the role of that?
So those mitochondrial reactive oxygen species can be involved in that inflammasome formation, or they may also be just sort of a general signal that the cell is under stress. So one of the things that they sort of do here that's kind of interesting is they use an inhibitor, MCC 950, to tell us that they are looking at an inflammasome that is going through a protein called NLRP3. So NLRP3, you could imagine, is, say, sensing the lipid. But if you actually look at what we know about NLRP3, it has been shown to sense a whole lot of different molecules that aren't really related. And if you think about it, it's kind of hard to imagine how one protein could sense all of those things. And what we're beginning to realize is that NLRP3 seems to be stressing or be sensing changes in ion concentration or changes of the plasma membrane permeability.
Right.
And so the mitochondrial reactive oxygen species may be part of that. They sort of hypothesize at one point here that the liposome could be altering the plasma membrane of the cell.
Right.
And so that could be part of how the liposome is actually inducing signal 2.
Right. So they think the liposomes, which they call them cationic liposomes, which and they're made up of lipids DOTMA and DOPE.
Yep.
Those are abbreviations for the lipids. They induce the reactive oxygen species,
and
that
also
gives
calcium changes.
And calcium changes as well, and that gives you signal 2, right?
Yeah, and so it may be that either the calcium or disruption of the plasma membrane and some other things that happen with like potassium ions could be what NLRP3 is sensing.
How cool is that? That here we have a RNA liposome that just happens to hit both signal 1 and signal 2, right?
Exactly. I have done some experiments where I have to think very carefully about what I'm using to hit signal 1 versus signal 2. And it's kind of nice here that they lucked out and got something to hit both.
So all of this is done in cells and culture. So they want to know, does this happen actually in people? So they have a cohort of patients who are being treated with this RNA LPX for cancer, 9 patients. And they've given them intravenously 25 micrograms of RNA LPX. It's encoding cevumeran, autogene cevumeran. Okay, so somehow that's helping them with their cancer. The idea is that it would help them. But the point is they give them this RNA LPX, and in all 9 patients, they see an increase in IL-1 beta in the blood.
Yes. So it looks like this is really happening in vivo.
Yeah. That's pretty cool. And they also see the same thing in mice. When they give mice, you see an upregulation. In this case, so they say it's IL-1.
Right.
So what's the difference? And also IL-1 alpha. So you told us about IL-1 beta. What's IL-1 alpha and IL-1? So
IL-1 is sort of a general term for anything that binds to the IL-1 receptor is kind of how I think of it. So IL-1 alpha is a different cytokine. It goes through a similar receptor to IL-1 beta, but it is often released upon cell stress. Some people talk about it as something called an alarmin, and it doesn't require these 2 signals. So it leads to some different effects on cells.
So they do some experiments to try and understand what's going on here. So and I think the one that I really like, if you add antibodies to IL-1 beta to these PBMCs in culture, and then you add your RNA LPX, you don't get the inflammatory cytokine production.
Yeah, I think that's really important. In the previous figure, so right now we're sort of talking about figure 2, but in figure 1, they had looked at cells in culture and they looked at cytokine production with sort of a modified ELISA. And when I looked at those data, IL-1 beta stood out, but it wasn't the number one standout cytokine to me. The cytokine that really stood out to me was IL-6 that was being made at really high levels, another inflammatory cytokine. And the great part about the experiment that Vincent just mentioned is that they show in this experiment that if you treat cells with an antibody against IL-1, then the IL-6 is not made. So the IL-6 is happening as a result of that IL-1.
And TNF is also not made. So both massive pro-inflammatory cytokines, you need IL-1 first before they get made, right?
Yes.
Yeah. Okay. So then they turn to mice who have been knocked out for the IL-1 receptor, right? IL-1ra.
Yes, exactly.
So that receptor, the IL-1 receptor, does that bind alpha and beta?
It can bind to alpha and beta, and as we will see later in the paper, it also binds to something else.
Okay. So in mice without the IL-1 receptor, if you give them RNA LPX, you get much less induction of systemic cytokines compared to wild-type mice. Now, we have to say the induction in wild-type mice is still lower than in human cells.
Yes,
right?
exactly.
And we're going to find out why.
Right. So
in a bit.
At this point, as I was reading the paper, I was a little bit confused because I had read the introduction and thought a lot about, I was going to learn some cool things about mouse versus humans. And at this point, I didn't think the mice and the humans were all that different.
Now, they also have NLRP3 knockout mice, which is the sensor for these RNA LPXs. And in those mice, when you give them RNA LPX, you don't have a decrease that you saw with IL-1 receptor knockout mice, right?
Yes, that is correct.
So they say IL-1 appears to have a determinative role in amplifying cytokines, which we understand. I don't understand the NLRP3 result. Can you explain that?
So basically, there are a number of different possible receptors that could lead to the inflammasome being turned on here. So some of this could be that you are seeing a different inflammasome, or some of this could be that in mice, you tend to see IL-1 alpha being made, which doesn't require the inflammasome.
Because in human cells, the inhibitor of NLRP3 did knock down IL-1 beta production, right?
Right, exactly. Yeah. And so here we're not looking at IL-1 beta, we're looking at IL-1 alpha.
Okay.
So you wouldn't expect NLRP3, or they talk about Gasdarmin, to really be playing a role, since that's an IL-1 beta specific thing, not an IL-1 alpha thing.
Okay. All right. Now, as Brianne already said, IL-1 is highly inflammatory.
Yes.
It's kind of like a politician. And so they say it has to be tightly regulated. There is soluble IL-1 receptor and membrane forms of what they call IL-1 receptor 2, which act as ligand traps. I presume IL receptor 2 is not transducing after it binds IL-1, right? Correct.
Yeah, it's basically
it's a
soaking
trap.
up. Yep.
So the soluble and the membrane-bound, they're traps to regulate, because, if you make a lot of IL-1, a burst, you have to tamp it down at some point. Otherwise, you have a cytokine storm.
Yeah, you want to soak up the excess.
So these are the regulators. And then they said, do these play any role in the difference between mice and humans?
Well, they did, but they also looked at one other thing here.
Yes.
Which is IL-1ra.
That's right. Oh, that's right. Another secreted cytokine, right?
Yeah, so this is sort of a third thing that could be secreted. You could secrete IL-1 alpha, IL-1 beta, or IL-1ra, and that is an anti-inflammatory cytokine. It's sort of a negative regulator or an antagonist of the other two. So
it binds IL-1 receptor 1, and it somehow downregulate the signaling?
So it gives you a negative signal and also competes with the active IL-1 signal.
All right. So they wanted to know if IL-1 RA was playing some role in this because they knew about this regulation, right?
Mm-hmm.
So they add RNA LPX to human PBMCs, and it measure IL-1 alpha, IL-1 beta, and IL-1ra released. Okay, at lower doses, less than 2 micrograms per mil, beta and RA were released equivalent amounts. At higher doses, there was 10 times more IL-1 beta, much more than IL-1ra.
Yeah, so it's sort of like the IL-1 beta is now, overcoming whatever
Right.
effect the IL-1ra can do. So they say
Right.
here the buffering capacity of IL-1ra is likely to be overcome.
And they use a phrase often. I don't know if you notice this. They write of note. Of note, IL-1 alpha levels remained low at all RNA LPX dose levels in human cells.
I actually didn't notice that, but I think that's actually a phrase I tend to overuse. So that might
you use
be why.
of note? Okay.
That might be why I didn't notice.
Okay, so mice are much less sensitive to RNA LPX than humans. So are similar things happening in mice? And Brianne, they write, in stark contrast.
In stark contrast.
Stark. Do you know George Stark? He was actually an innate immunologist.
I don't know George Stark.
The word, look it up. Yeah, he was an early interferon guy. So at baseline, you already have a lot of IL-1RA in murine white blood cells. And then when you add RNA LPX, it goes up even more. And so you don't see much IL-1 alpha and beta except at high dose levels of the RNA LPX.
And even when you do get that high amount of IL-1 beta and IL-1 alpha, or at least an induced amount of those two,
right.
IL-1RA is also further induced. And so even at that point, it's in 100-fold molar excess over the IL-1 beta and 10-fold excess over IL-1 alpha.
Right.
And they do this really nice experiment where they show all of the all 3 products normalized to what they were at baseline. And so you can see that you do get some induction of IL-1 beta and IL-1 alpha, but in mouse, you get this dramatic induction of IL-1RA as well that you don't see in human. And IL-1RA is the negative regulator.
Wait a minute. I skipped ahead too far. Hang on. Okay. Where was I? 2P release in vivo. Oh. I went way ahead. Sorry, folks. Okay.
This was really exciting, and it makes me want to go look at some different stimuli that I have in my lab, where I often look at IL-1 beta release, and now I want to know what happens with IL-1RA release in those same situations.
So they treated mice also with RNA LPX. So the experiment we described was mouse white blood cells. So they did mice, and they also see upregulation of IL-1 beta and alpha, and also high levels of IL-1A in, as you said, I think 100-fold molar excess over beta, 10-fold over alpha. Yes, you already said that, right? And they did this in several strains of mice, and it was the same in all of them, right?
Right.
Which is cool. Yeah. Okay, so what about humans and RA? So human patients treated with 25 micrograms of RNA LPX, they have a slight increase of IL-1 beta over 1A, whereas in mice, much more RA compared to 1 beta. So big difference in humans versus mice. And then they say of note again. They conclude IL-1RA can attenuate the effect of 1 beta in humans, but only at low to moderate doses of RNA LPX. Whereas the substantial induction of RA in mice gives you a higher degree of what they call attenuation against increases of alpha beta. And so the mechanism of that would be interesting to sort out, right?
It would. I also really like that they have this one other piece of extended data here, because my brain went straight to another set of studies at exactly this point. And then they addressed exactly my question really nicely here, which is part of me wondered, well, is any of this related to the difference in mice being in more pathogen-free conditions or more clean conditions and humans being around microbes? Maybe, human cells are just ready to be inflamed because we spend so much time with microbes. And mice, we keep in a clean environment. So that's what's going on here. So they also looked at non-human primates cells.
Yes.
And you would imagine that non-human primates are also kept in facilities that are more like where humans live. And of course, are also evolutionarily closer to humans. And so if my hypothesis that this was all about sort of microbial exposure or cleanliness or whatever you want to call it was correct, then we would say, then the non-human primates would look like people. But in fact, they found that the non-human primates had responses that were similar to those responses of mice.
Right.
Both telling us reasons why we might not have seen this preclinically in non-human primate studies of vaccines, and sort of shooting down my hypothesis. And I was so glad that they did that.
So the conclusion is non-human primates might not fully capture the inflammatory toxicities related to RNA vaccines. Very interesting.
Yes.
I would have said NHPs were like people, right?
Right. That's what I would have expected as well.
Yeah. Funny. Okay. So is IL-1ra the reason for the difference between humans and mice? So they give RNA LPX to IL-1ra knockout mice. So this is now the first knockout of this particular gene that we've seen.
Yeah. So now we're knocking out this negative regulator, not one of the inflammatory cytokines.
And what would you have predicted, Brianne?
I guess that I would have predicted that if we got rid of the IL-1 receptor alpha, then perhaps we would see the mice looking more like humans.
Yes, exactly what they see. They treat them, they rapidly develop a cytokine release syndrome-like phenotype, pronounced hypothermia, body weight loss, excessive systemic cytokine release. But they were transient and they resolved on their own, which happens in people, right? You get your mRNA vaccine, you have some of these things, and you're okay in a couple of days.
Yeah, the body weight change was impressive.
Yes.
The mice lost 10% of their body weight in 24 hours. That seemed like a lot more than I would have expected that quickly.
Yeah. They see a lot of pro-inflammatory cytokines being induced in the absence of this regulator, IL-1ra. So it seems that that's the key there, right?
It is.
They did a few experiments to understand the contribution of the myeloid compartment on this response. So they have their IL-1RN null mice. They give them a tyrosine kinase, a FMS-like tyrosine kinase 3 ligand, which they say induces the expansion of splenic macrophages, monocytes, dendritic cells, and neutrophils. So you're making more of these cells.
Yes, for immunology aficionados, it's the FLT3 ligand. And
so these are all myeloid-derived lineage cells, right?
Yes.
Yes, FLT3 ligand. I'm not used to saying it. I was once at a meeting and I read out the whole thing, and someone just said, "Yeah, FLT3." So when you amplify all these cell types, you get an enhanced cytokine response, even in these IL-1RN null mice, because now you've amplified all these cells. But they don't see it in wild type animals that are pretreated with the same FLT3 ligand. Maybe because they make a lot of IL-1RA.
Yeah, that's what I assumed, is that we're just making so much IL-1RA that it is able to turn down even the excessive myeloid response.
So they conclude that this IL-1ra is actually suppressing the IL-1 induced adverse events in mice and probably explains why they're resistant to high doses of RNA LPX. Okay. They do some experiments with T cells now, Brianne. You want to know so IL-1 is involved in development of adaptive immunity, and they wanted to know.
Yeah, the innate immune response is really important for helping turn on an appropriate adaptive immune response. And so another great part of IL-1 is that when you make a lot of IL-1, that helps you induce a good T cell response. So perhaps all of the IL-1 we see with these vaccines leads to the T cell responses that we talked about in the first paper.
What's the mechanism of IL-1 and T cell responses? Do we know?
It is kind of thought of as helping co-stimulation.
Okay. So this is happening in a lymph node.
Yeah.
Okay. So where an APC is presenting antigen to a T cell, you got some IL-1 around, you're going to have a better...
You're going to better activate that T cell.
Activate it. Okay. So they inject wild type or these IL-1RN null mice with RNA LPX. They see expansion of neoantigen-specific T cells in both genotypes, but higher T cell counts in IL-1RA deficient mice compared with wild-type mice. And they look at the quality of these T cells. They have memory markers, but in the RA knockout mice, is that correct? Yes. They have RA deficient mice. Those were short-lived effector cells, more of those than memory cells. And so they say, they conclude that unmodified RNA vaccines are a good way to make antigen-specific T cells when you have enough of innate stimulus. Did I get that right?
Yeah. So basically what they're showing is that this excess amount, or I don't know if excess is the right word, this increase in IL-1RA in mice is limiting the T cell response a bit.
Right.
And if you get rid of that, then you see a better T cell response.
Okay.
So it's sort of like showing you the pros and cons.
So we must be getting a good T cell response, right?
So potentially this could mean that we're giving a good T cell response.
Yeah,
From earlier parts of the paper, you might say, boy, I wish that I had more IL-1RA, so I didn't have so many side effects from the vaccine. But then this might make you say, oh, wait, maybe I don't want too much because I would like to have a good T cell response.
That's cool. And I wonder why it evolved that way, right?
Right. Yeah. That I think is fascinating, and trying to figure out why these types of things evolved is so cool.
I mean, it must have to do with the different biology of mice and humans, the different environments that they live in and are subject to selection in.
Yes. We know that there are some PRR differences between the two species. And I assume that they have to do with the environment and the particular microbes that are infecting each.
So there are other ways to induce pro-inflammatory cytokines besides the NLRP3 that we've talked about. And they actually do a series of experiments where they look at that. And it turns out that IL-1RA can also protect mice against excess inflammation triggered by a variety of innate stimulatory agents. The part I was really interested in was they said they looked at CAR T cell and tumor-directed antibodies, bispecific antibodies, because they said those can cause cytokine release syndrome in people, right? CAR T cells, which are directed at a tumor, or tumor-directed antibodies can cause that. So,
right, and they actually had mentioned this earlier in the paper as well. I'm trying to find where that was, because I think that this is a sort of key point. I don't know where it was, so I guess you can keep going.
So these immunotherapies trigger induction of IL-1 beta by activating T cells. rather than a primary release caused by the sensing of RNA LPX. So they said, if this IL-1 beta contributes to the development of this cytokine release syndrome in IL-1RN null mice,
right?
Which have the tumors that are targets for the therapy, right?
Right. So yes, exactly. And I found earlier when they were doing those experiments where they used the IL-1 antibodies to block and showed that they could not get IL-6 production.
Right.
They also looked at what happened with blocking TNF and IL-6, and they didn't see as much of an effect there. They said that most of these inflammatory cytokines were coming through the IL-1 beta pathway. And they note that that is really unique from what's happening in T-cell activating therapies.
Right.
So the reason why this part of the paper is important is that here they're saying, okay, well, can IL-1ra help even in the case of cytokine release that's coming through kind of a completely different pathway?
And it does, right?
Related. Yeah, and it does.
Yeah. And so in these experiments, they find that RA does attenuate the inflammatory response to these particular therapies in mice. So this is important because in people you have these issues, and maybe this gives you an idea for some kind of therapy to prevent it, right?
Yeah, absolutely. I, hadn't thought about IL-1RA as much before this paper. Yeah. And this paper kind of really makes it very clear that this molecule is rather broadly important for limiting innate immune responses, and that maybe we're underestimating it, and it can play a role in excess cytokines or inflammatory cytokine release in even diseases where we might not have otherwise thought it would be relevant.
So if you take away in these IL-1RN null mice, they still can handle, the RNA LPX, and they find that that's because they upregulate the production of IL-1R1. They actually shed it from cells, right? So that's the receptor that would be functional, but there's a shed version that's a sink. And they say, in the absence of IL-1RN, this is increased, and that compensates to a certain extent for it, right?
Yes.
It's a soluble sink for IL-1 alpha beta in the absence of IL-1RA. So IL-1RA encoded by IL-1RN gene, is that correct?
Yes, that's correct.
Because, that when you take that away, IL-1R1 comes to the rescue. Isn't that interesting?
Yeah. So it also shows us just how important it is to regulate the IL-1 levels. Because we have these redundant ways of doing so.
Oh, so the last set of experiments I think are very cool. They have to do with the lipid. So remember, RNA LPX is the liposome unmodified RNA. And that's different from the vaccines, the COVID vaccines, right? They're modified RNA with lipid nanoparticles, which are different chemical composition. So remember, RNA LPX has two signals that it can use to turn on these events that we've talked about. The RNA vaccines, they still elicit, these inflammatory signals in patients after intramuscular inoculation. And they are formulated, instead of cationic liposomes, they have ionizable lipids, which they say provide structural stability to the particle, and they were designed to allow the particles to get out of the endosome, which they have to do to be translated so the RNA can be translated into cytoplasm.
And they do note that it is presumably because some of the details of this are challenging to understand.
It was an empirical develment, right?
Yes, it was.
Empirically said, let's see what makes better translation. So that's the assumption there. So they want to know what's different about the induction of IL-1 family members by these mod RNA LNP. So previously we were talking about RNA LPX, right? Unmodified RNA liposome. Now we're talking about modified RNAs in a LNP, lipid nanoparticle. They use two different ionizable lipids to make these. MC3, which are often used for packaging small interfering RNAs, or SM102. That's what is in the Moderna vaccine.
Yeah, and that confused me because this group of authors is from BioNTech.
Isn't that funny? Yes.
And they're using the lipid. They're not using the Pfizer lipid. They're using the Moderna lipid.
I don't know why. That's a good question. I don't know why that was done. Yeah. They're doing the experiments with the other company's vaccine. So now they have human PBMCs in culture. They add these, and they see, again, just like for RNA LPX, you see an increased IL-1 beta produced with either mod RNA LNP formulation, with either lipid. And IL-1 alpha and IL-1ra remain low, just like for the RNA LPX, right?
Mm-hmm.
And so they do a variety of combinations of the lipids, empty and so forth. And they conclude that these lipids can play different roles by either providing signal 2 or signals 1 and 2. So signal 2 is provided by MC3 lipid, and signals 1 and 2 together by SM102. So SM102 can provide both signals. Isn't that interesting?
That is really interesting. And it's not going to give you a antigen-specific response, obviously, because you don't have the antigen in it. But by itself, it's going to give you a nice innate response.
And it would have to, because the RNA is modified. So that's going to be reduced, right? So it makes sense that the lipid, which is used in the modified RNA Moderna vaccine, is providing both signals, which explains the induction of IL-1. Right?
And the side effects that potentially are seen.
So then they use the modified RNA with liposome, okay, which is not what any of us got, but this is an experimental, and compared the cytokines with RNA LPX and mod RNA LNP with SM102. So that's the Moderna vaccine, mod RNA LNP SM102. You have RNA LPX, which we've talked about a lot. And if you're still with us, good for you. This is very complicated. And then they have
cool
mod RNA LPX. Okay. What happened? And again, these are with PBMCs in culture. Now, the RNA LPX gives you cytokines, as we showed before. Mod RNA LPX did not induce IL-1 beta, and consequently, none of the downstream cytokines, TNF, IL-6, right? I guess those would be some of the downstream ones. But if you use mod RNA LNP SM102, you get the same cytokine induction profile that you see with RNA LPX.
Right. And so what this tells us is that the lipid matters. It's not just about the RNA and the RNA vaccine. The specific lipid that you use can influence how much IL-1 beta and innate immune signaling you have, which could influence your side effects. But we also saw earlier that the IL-1 beta can impact the T cell response. And so that lipid can be giving you a lot of different issues or a lot of different changes in the vaccine outcome.
So they put it this way: reactogenicity of modified RNA is context dependent. Mod RNA, so the modified RNA, if you put that in a liposome, it's not stimulatory. It doesn't stimulate much of a IL-1 response because you don't have both signals, right?
Mm-hmm.
But the mod RNA, if you formulate in an LNP with MC3 lipid, it's weakly immunostimulatory. And now if you put a mod RNA in an LNP with SM102, it gives you a potent inflammatory response. And that's, of course, the Moderna vaccine. And it partly explains why it works so well, right?
Yeah,
it's
exactly.
Just a great combination. But I think what we learned here is that we didn't predict the reactogenicity in people because the mice and the non-human primates, we can give them a lot and it's okay.
Right. And I think that is also just really key to know, because now we know something else we should be looking for in developing vaccines like this. We need to be potentially looking at IL-1ra And we have an idea of why things might look different in other organisms.
So one thing from the discussion I found very interesting, there is a recombinant IL-1ra, anakinra.
Yes.
Great name.
Yes.
And they suggested it might be useful for treating people with cytokine release COVID, right?
Right. I feel like that was tried.
Yeah.
I thought it was. I could be mistaken.
Yeah, they do some references here.
But I thought it was tried. Because I remember thinking that it was going to be a great idea and being disappointed.
Because according to what they showed, you would think, right?
Right.
But maybe it's too late by the time you have the actual severe disease, right?
In some ways it also makes you think, well, would anakinra be helpful with the vaccine to reduce your side effects? But would that reduce your T-cell response?
Yeah. Another point which we've made, it's worth emphasizing, that it's not just the nucleic acid that's important, but it's the lipid. The lipids matter. Mm-hmm. They both determine the cytokine induction profile. And while it's, you have some side effects, it's good. It's good because that leads to robust antibody and T-cell responses. What else did I want to point out? They have great figures in this paper, great pictures of their their RNA lipid complexes. Okay, here's the last sentence, the last paragraph. IL-1ra deficient mice can better predict patient responses. So that's it.
Yes.
So now in the future, if you want to test a new RNA vaccine, you should use IL-1ra deficient mice, right?
Right, exactly.
To better predict patient responses. And they say, evolution has shaped and rewired how positive and negative regulators of IL-1R1 signaling are released. And these determine the magnitude of responses to RNA vaccines in mice, primates, non-human primates, and humans. But I think, as we've pointed out, that would be really interesting to see what were the evolutionary forces that did this and the selection.
Yeah, absolutely. I can imagine a lot of really interesting basic immunology coming out after this paper. I can see lots of questions to be asked.
The mechanism that keeps our IL-1ra high in mice, right? What's doing that?
Exactly.
It seems to me it's probably a promoter-related thing, right? It could be relatively straightforward to sort out.
Yeah, I mean, it could be relatively straightforward, because it's probably a promoter thing. I'm sort of more interested in why it's induced so highly in mice, as opposed to inducing IL-1 beta so highly.
Right. Anyway, very exciting paper. I thought that was just brilliant. Well done. Nicely written. The kind of COVID science I like. I knew you would like it.
Oh, I really liked it.
When I saw you on the schedule, I said, I'm going to do these. We're going to do these papers today.
Yeah, basically, you picked a paper that is related to my PhD work and another paper that was related to my postdoc work. So I was pretty happy.
There you go. We got you covered. Let's do a little bit of email, Brianne.
Okay.
Why don't you take that first one?
Sure. Agnes writes, Dear heroes of TWiV and Microbe TV, I am a longtime listener, and I wanted a reason to share my admiration for you and the education you provide. While this is my main reason, I also wanted to include a gentle correction on what Rich Condit said about oxidation and reduction in TWiV 873. I believe he mentioned reduction of primary alcohols to aldehyde. The first time I heard it, my chemistry senses tingled, and I played it back for confirmation. When I think of reduction with my students, I think of adding a hydrogen to an aldehyde to become a primary alcohol. For people who are not always with chemistry, that was the opposite. You can also look at it mechanistically and imagine an aluminum hydride donating a negatively charged hydrogen, also known as a hydride, to the carbonyl carbon of an aldehyde. This changes the CO double bond into a single bond, and a negative charge resides on the oxygen. The resulting alkoxide ion becomes a strong enough nucleophile to donate its electron pair on the oxygen to a hydrogen on another molecule, like an alcohol, such as ethanol if it is a solvent, to produce the resulting primary alcohol product. There is more to this, but at least at this level, it makes sense to me. Hope it helps. Please accept my deepest appreciation and gratitude for your valuable knowledge. When Rich and Kathy mention for the uninitiated listeners, I feel they are speaking directly to me. They, as well as all of the TWiV Microbe TV team, have opened up new worlds of learning for me. I only know chemistry, having received my doctorate in inorganic chemistry, with emphasis on crystallography. Hence, when you described the structure of Mimi virus's nucleocapsid, I was silently saying, more, I want to hear more. Absolutely loved it. Sincere apologies for the length of this. Feel free to edit. I wanted a chance to thank you all and contribute my 2 cents. From Agnes, and Agnes is at the Medical Sciences Center in Madison, Wisconsin.
Thank you. And hopefully Rich will hear.
Yes, hopefully he will.
I'm sure he knew that. Deborah writes, Dear Vincent and friends, thank you so much for all the education you've given me over the course of the pandemic. As someone who lived through SARS in China and in my role at the time at the U.S. Embassy, helped the CDC with its deployment, it never occurred to me that we would be so unable to implement basic preventive measures. After all, it was U.S. CDC that worked with their Chinese counterparts on track and trace. The Chinese have since become masters of it, and it turns out we were never able to do it ourselves. Listening and learning from you about what is working and not has been incredibly helpful. I wanted to address a few things from your most recent episode. As someone who lived in China for 18 years and specializes in Chinese environmental politics, firstly, the use of game animals as a term related to China is what the authors of the first paper say it is. It is about animals that are basically captured from the wild and, while generally farm raised for all or part of their lives, are not fully domesticated. You all then digress into hunting and African bushmeat and U.S. deer and elk and the like. By the way, that was Dixon. Hunting as such doesn't have anything to do with wild animal trade in China. For one thing, no one has any guns. Oh my gosh, what a great place. In my 18 years in China, I once met a German aid worker who said he went out and hunted wild boar and pheasant for his food, but that was incredibly unusual, and I've never heard about it otherwise. There is a bit of touristic stuff, but it doesn't exist as a way to enter the markets and the food industry. Thus, all of these game animals spend considerable time in captivity, which I think is an important part of the specific risk of ill health. Yes. Dixon likes to go off about various animals, yes, he I don't think he quite got the distinction, but
right.
Thank you for
the only thing that is important there is to say that eating more unique animals is not something that happens only in one area of the world, and we shouldn't stigmatize people because of it.
I think that was his main point, that people everywhere eat all sorts of things, and yeah, don't just pick on one. As for the taste for game meat, you're indeed correct that there are all kinds of cultural preferences for food. Yeah, that's what we were talking about. My impression is that there are cultures such as ours that are interested in trying new spices or flavors with the same proteins. And there are cultures like China's that are interested in new proteins, but generally cook them the same way. Nowadays, you do find people in China interested in trying Indian or Italian food, but more commonly, you find people who would like to try some new variety of meat or fish, but would prefer it cooked in the same way as other meats and fish. I'd also note that the U.S. is at the extreme end for limiting meat types and becoming more so all the time. I don't think I'm alone in having had grandparents who ate tripe and liver, which I don't eat. Yeah, I did too. I would never touch that stuff.
No.
His tripe stomach?
Yes, cow stomach.
So my grandmother used to make that, and she also made lung.
Whoa.
Oh my God. No. Chopped up lung and liver. My grandfather was crazy about liver. I don't eat any of them. And earlier generations of Americans ate things like rabbit. Yeah, the Italians still eat rabbit for sure, which is now quite rare. Sorry, no pun intended. But please call these markets or farmers markets. Wet market is a mistranslation of the Cantonese, not the language of mainland China, and seems to give people the impression these markets are different in some fundamental way from what Dixon described seeing in Thailand. They aren't. They are a pretty common type of market all over the world. And the main issue about China is how big it is and how large the animal husbandry market is. I don't know where the term wet market came from. I certainly didn't make it up. I just continued to use it. I think they call it wet because the animals are slaughtered, and so it's wet, right?
Yeah, I would be interested to know. The writer says that it's a mistranslation of the Cantonese.
Interesting.
So I'd like to know what the correct translation is.
Yes, Deborah must know. Maybe she speaks Chinese or Cantonese or whatever. We have chicken markets here in New York.
Right.
And they're not called wet markets. They're just called chicken markets. So you could call them meat markets, but the Huanan, it was also have fish, right? So Anyway, and she continues, as for why people purchase live animals, it is generally because they think it's a way to reduce the risk of getting bad meat, to prevent receiving an ill animal not to cause illness. I've never bought live meat, but I did often go to fish markets to choose fish out of a tank when I lived in China. I may be totally wrong about the reality, but my impression and those of my neighbors was that it was easier to tell if a fish was healthy if it was still swimming around. Look at how it swam. So I think that is what people are doing. They want their meat fresh and healthy. Obviously, it turns out in crowded cities not to be the greatest idea, but the motivation isn't hard to fathom for a country that does not trust its food safety regulators and does have a huge problem with food adulteration. Yeah, it's interesting, isn't it, that I could see that.
Yeah, I can totally see that. And I hadn't thought about that before, but that makes sense as reasoning for why that would be your preference for where to get your meat.
I wonder, though, if the people who frequent these markets really know the risk associated with them.
Yeah, one assumes that they probably do not know that risk.
Yeah, I would assume so. Just thought I'd share a bit. I found listening to your discussions of papers incredibly illuminating, Deborah. I thank you, Deborah. It's wonderful. I appreciate viewpoints from people who are there because I've never been to China. I just relate what I read. So
yeah, I think that Deborah brings up a lot of really useful points that help me think about this process.
Brianne, can you take the next one?
Sure. Brenda writes, TWiV 876, what an interesting podcast. A few thoughts. Like you translate degrees centigrade to Fahrenheit, kilometers need to be paired up with miles. I must confess that I had to do some Googling of various walking distances in my vicinity to get a handle on 1.1 kilometers. Was any study done on the workers in the wet market, especially in the wildlife trade and nearby stalls? Where they live might be interesting. It might connect the cases with no known direct connection to the market. When I lived in an apartment building, I could tell you where my immediate neighbors worked, but not the rest. That would be 2 out of 60 units sharing one elevator. What is the likelihood of the wildlife traders getting wind of the impending closure and clearing out the illegal animals before the local officials got there? From my experience, I would suggest chances are that is how it played out. And Brenda is from Black Isle, Scotland. I remember that we talked about a paper a while ago that looked at diversity of animals in a similar market a bit before that had some trust with the sellers, that they were just sort of getting a overall idea of which animals were there. And they didn't, if I remember correctly, talk about many of the illegal animals being there either. But I could assume that it's certainly possible that some of them were cleared out.
As for the locations where they live, I don't know. I mean, Michael might have found those data, right? He was combing them quite closely. He found other data that weren't in the WHO report. That's part of his paper. So maybe you could find some of it there because you probably have access to the raw data.
Right.
Kate writes, on episode 876, Spillover Market with Michael Warraby, the guest was asked if people around the market could give serum samples for testing. He said at this point they would be corrupted. And what he meant was that people would have caught COVID, right, would have had COVID, and therefore you wouldn't know when they were.
COVID or been vaccinated or something.
Yes. I was wondering if the zero COVID policy in China might mean you could still get some good samples. Assuming the virus got contained and maybe some people didn't get a vaccine, couldn't you still attempt to research this population? I don't know how you would tell the difference still. So you would have, you want to have seropositive people, right?
Right. And it's possible that the antibody levels are going to decrease over time. And so it would be hard to get the seropositives. In China, they've been using the Sinovac vaccine, which is an inactivated. And so it should give some immune responses to both S and N antigens. So here we can do things where we can test for immune responses against S and know that those could have come from vaccine or from infection. And we can also look at responses against N and know those only came from infection. And so we can sort of distinguish. With the inactivated, you can't do that distinction. I just don't know how you could ever have certainty about your data.
We have one more, Brianne.
All right. John writes, Doctors TWiV, last throes of winter here in Greater Braddock, just like you're experiencing. I made a few loops around the back acre on my cross-country skis just to be able to say that I had. Consumption of critters. When I was a grad student at Rutgers, for trips back home to the DC area, we only ever took I-95 once. Forever after, we'd take the back route down Route 1 and US 13 in Delaware and onto 301 through the Eastern Shore to 50 and then across the Chesapeake. There was a place we'd pass on 13 that always had a sign by their mailbox, oven-ready ducks, muskrats. I never had a chance to experience the place since I was never tempted to try a muskrat, and my ever-impatient future ex never wanted to stop for a duck. But I regret that I never managed it. But if someone were to come up with a way to prepare groundhog that would make it palatable to the point of being hunted to extinction, you wouldn't find me complaining. Then, with the discovery of the Endurance, which of course is enormously cool, a COVID reference. My old W&M, perhaps William and Mary, classmate Jerry Coyne, who may be familiar to you, is currently returning to Chile on a Norwegian ship that just passed close to the site of the discovery. One of the crew on his ship knows one of those on board the Discovery ship. Jerry would still be down in Antarctica, except that despite the tightest of COVID testing prior to boarding and then afterward while at sea, several passengers came down with COVID a few days after they were at sea, and the remainder of the voyage was scrubbed. It ain't over. Ari, the Endurance, you mentioned the Vasa, which I visited many times, but there's another beautifully preserved Swedish shipwreck discovered more recently that you might not be aware of. The Erik Nordvall was a sidewheel steamboat built to ply the newly completed Jurta Canal, built to circumvent Danish taxation of ships sailing around the southern coast. The tax was dropped 3 weeks after the canal opened. That connected Stockholm with Gothenburg, Göteborg, aka Gothenburg. By taking advantage of two large lakes in between, Vättern and Vänern. Things went well with Erik Nordvall for 19 years until 1856, when it foundered on a reef in Lake Vättern, and then abruptly sank no sooner than it had been towed free. If that had not happened, the Erik Nordvall would have long since disappeared from any memory, just like its four sister ships. But while it disappeared from the surface, it didn't vanish. It stayed upright on the bottom, even better preserved than the Vasa. You can read the fairly succinct story of its past and its discovery here. The site is a little weird. Just scroll down past the images to get to the text. And he gives a link. But what did they do next? They could raise it or reconstruct it. They chose the latter, using it as a training exercise for traditional wooden shipbuilding in scrupulously authentic detail, and even fitted it with precise replicas of the original wood-fired steam engines actually built in the same factory as the original ones in the 1830s. That ship, the Erik Nordvall II, christened by Crown Princess Victoria, now plies the same Jurta Canal in the summer months as a tourist attraction. If you simply search Erik Nordvall II, you'll get numerous YouTubes of it, as well as articles about it, including this one in the journal Industrial Archaeology. Who knew there was such a journal? And he gives another link. Since the last half of this is all about Sweden, I'll close with a typical Swedish closing. Med vänliga hälsningar, with friendly greetings. The world could use more of that attitude. From John.
Wow, this ship, if you go to this site, it's just sitting beautifully preserved on the bottom.
It's so cool.
Look at it.
Look
Isn't
at that.
that cool?
Yeah.
Looks like there's some ropes.
That you can see.
Wow, and the mast is covered with barnacles, I guess.
Yeah, I guess.
Looks like someone rearranged things, right? It's so clean. Wow,
yeah.
That's beautiful. Wonder how far down that is. If people dive down there.
Dive?
You know?
I don't know.
Anyway, it's cool. They reconstructed it and made a ship that's sailing. Nice. Thank you, John.
Yeah, that's cool.
I love the pronunciation. I didn't know. So, Göteborg, right? I always say Gothenburg, right?
Yes, exactly.
And I know that the Swedish is, well, I know how it's spelled, right? I always pronounce it Göteborg, but it's Göteborg.
I never would have come up with Göteborg.
It's probably Göteborg, I bet.
Yes.
I don't know. I did a TWiV in Sweden with some, Swedish scientists, and they're trying to teach me how to pronounce their name, and I was just not good at it. Not good at it. All right. Some pick time, Brianne. What do you have this week?
I have an article that I read recently that really surprised me. So it is called COVID Restrictions Prevented Dengue in Hundreds of Thousands of People in 2020. So this is a New York Times article from earlier in March that reports on a study that came out in The Lancet looking at dengue infections in April 2020. And one sort of expected that perhaps dengue either wouldn't be changed with COVID restrictions. That was perhaps what I would have expected. Or that, if anything, perhaps there might be somewhat more dengue because some of the mitigation measures, like spraying for mosquitoes, went away. Mm-hmm. And instead, these authors found a large decline in dengue cases during 2020, which is great, given the high risk of dengue in many individuals, but also led these authors to try to figure out why did dengue numbers seem to change during 2020. Wearing a mask is not going to change your likelihood of getting bit by a mosquito, for example. Like it might change, your flu susceptibility. And what they were able to find was that people staying home, particularly the disruption in movement, seemed to be the only explanation that seemed to fit the data. In particular, they thought that school closures seemed to be a big part of cutting down the dengue cases. And they realized that this made sense because the vector, Aedes aegypti, is a daytime feeding mosquito. And so it is likely going to bite people when they are at their locations in the daytime, like school or work. And most mosquito control programs focus on people's houses, not public places where people might be during the day. And so they realized that changed susceptibility to dengue. And that gave them some new ideas about dengue transmission and ways that you might combat dengue. So maybe spraying around houses is less important than spraying near workplaces and schools where people spend time during the day with a daytime biting mosquito. It seems so obvious in reading it, but I never would have expected it. And it's one of those ways that this pandemic is giving us some unexpected virology knowledge.
So basically, the lockdown, people are home, and the homes were still sprayed for
Right.
mosquitoes?
The homes were still sprayed.
Okay.
And they weren't going to schools or workplaces that weren't sprayed.
That's great. That's, I think, epidemiology at its best.
Yeah,
right?
I think so too. And so they then talk about the fact that they don't yet have the data for 2021, but they're worried that the numbers might come back up now because of reduced spraying.
Of course. And also people going back out again.
Exactly,
right?
yes.
Yeah. Cool. That's very cool. I have a different pick. I want to go get it, so I'll be right back. Well, unfortunately, I can't find it.
Oh, no.
This is such a useful thing. What did I do with it? It's not such a big place. Anyway, this is a funny pick, but I love this stuff so much. It's Scotch Dual Lock Fastener. So I'll put a link. You can go look at it. And this is basically a strip. So you know what dual lock fastener is? It has these, it's kind of like Velcro, but it's way stronger than Velcro. All right. It's a strip with one side is a peel off paper that you can use it to stick to things. And the other side has the little projection sticking up, and you cut two of these pieces and you snap them together and they're not coming apart. You have to pull really hard. So you can use it to put things places. So and my table that I'm sitting at here, there's a lot of stuff underneath it that I've attached. Like there's an outlet strip that I use this to attach it. I have the Ethernet switch underneath. I used to have my Mac Mini hanging from it's so strong. It is perfect.
That sounds great.
It can come in different widths. I used it to put the mailbox on the door here, for example, and it's not going anywhere. So it's very, very strong. But when you snap it together, those tines, they just snap into each other. And I tried Velcro. You can buy Velcro, but it doesn't have nearly the strength of this. So it's a little pricier than Velcro. A 10-foot roll at Amazon is $22 of an inch wide, and I think it comes in different widths. You don't need more than an inch. You can put multiple strips on things that you need to hang up. For example, the Mac Mini, which is a pound or two, I had a 3-inch strip on either side, and it just had no problem hanging up there. So if you need to hang anything within reason, it's not going to hold 50 pounds, but I'd say anything less than 10 pounds or so, you should try this. I even have things on my desk that I don't want to move around anchored down, right, with this. So this has been so useful here and at home. I wanted to share it with you. So it's called Scotch. It's got a certain number, 3M Scotch Dual Lock Fastener. Just one of those things.
It sounds like a lot of people who are in the lab would find that useful in the lab.
Oh, absolutely. You could put your holder for pipette men, right?
Mm-hmm, right.
It usually comes with double stick tape that eventually falls off. You could put this on, it would never fall off.
Right.
It would never fall off. Yeah. You could put all kinds of things up in a lab with this as well. Yeah. So I needed to hang things in various places, particularly underneath the desk, right?
Right.
And it was perfect. I didn't find it on my own. I have to admit, I did find it on a YouTube channel. A guy was hanging computer monitors on the wall, right? And he was using this. They're hanging up. They're not going anywhere. And so I said, I have to try that.
So it can hold a lot then.
It can hold quite a bit, yeah. It can hold quite a bit. Very useful stuff. And I'm sure you could find all kinds of uses. The only thing is, if you want to take it off, it takes quite a bit of pulling to get it off, and you need to be able to get a grip on whatever you're sticking up. So just but most of the stuff that I'm putting up, I don't need to get it off, so it's fine. But I think it's incredibly useful. So check it out. We have a listener pick from Aaron, who writes, "Hello, TWiV team. I wanted to let you know that I love how closely you're following the James Webb Space Telescope, and though I might like to know that, like to know that I'm both a longtime TWiV listener, I won one of your Drobo giveaways in 2010. Wow. And also a microbiologist turned engineer who worked directly on telescope before it launched. I studied virology in undergrad and grad school, but through a series of unexpected connections, ended up working for NASA as a planetary protection engineer and contamination control engineer. Anyone who has worked in a microbiology lab knows that contamination can ruin your data. And it turns out that contamination on a spacecraft science instrument can also ruin the data. And once it's launched, we usually can't go up and clean it up. I work in NASA clean rooms to develop strategies to maintain, monitor, and verify spacecraft cleanliness levels. Not all spacecraft have to be microbially clean, but anything we send to Mars or icy moons like Europa does. The James Webb Space Telescope was particularly sensitive to particulate or molecular contamination, so that was my focus when I worked on that mission. I don't do much virology anymore, but viruses are still my first love, and I use TWiV to stay in touch with what is happening in the world of virology. Thank you especially for your work during the pandemic. I have recommended your podcast more times than I can remember, and even more frequently found myself sharing things that I learned on the podcast with friends and family that had questions about the virus. In case you're wondering what a former virologist does in NASA clean rooms, I have 2 video pics. The first one was produced just before JWST was launched, and there's a segment starting around 6:40 talking about why JWST needed to be so clean and what contamination control engineers did to make that possible. I'm not in this video, but some of my coworkers are. The second is a clip that shows how we monitor microorganisms on hardware that we send to places like Mars or the icy moon Europa. Exploration Outer Space, Season 5, Episode 5, starting at 5:55. The show visits the Goddard Space Flight Center clean room, where I take samples from an electronic box that will one day land on Mars. Cool. Thanks again,
cool.
Erin. So I remember Erin because I was at ASV early a number of years ago, and she came up to me and said, "Hi, I'm Erin. I won the Drobo." And I said, "Oh my gosh, nice to meet you." And so I know we met, and it's great that you segued into this incredible career. Good for you.
That the video about how you try to prepare the things that are going to the icy moons like Europa or Mars sounds so cool, and I can't wait to go read it or to go watch it. And I also just really love the title Planetary Protection Engineer, Erin. So know that I'm a little jealous that you get to say that that's your title.
Erin, maybe you should come on one of our pods and talk about it in more detail. Maybe not TWiV because it's bacteria you're largely concerned with. Maybe we could bring you on TWiV. Anyway, you know how to get in touch if you're interested. That'll do it for TWiV 882. You can find the show notes at microbe.tv/TWiV. You can send your questions, comments, pics to twiv@microbe.tv. And if you like what we do, consider supporting us financially. You can go to microbe.tv/contribute. Microbe TV is a 501(c), which means your contributions are tax deductible in the U.S. Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. These were some really fun papers.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.